Inside a human brain, there are many different types of cells, kind of like the different pieces of fruit inside this fruit cake. We can't know the shape, size and color of each fruit piece in the cake just by looking at a slice. For that, we would need to sort them out into separate piles. Similarly, it's useful to separate out the brain cells of different types and study them one at a time. Some brains at our brain bank have been frozen at -80 degrees Celsius for over 20 years, so they are quite fragile. Just as it can be tedious to pick out fruit pieces from a cake, separating out the individual nuclei or inner cores of the cells from our brain samples is very challenging. We break down the tissue using glass tubes and pestles, filter the sample through a fine sieve, and wash it with solutions to remove debris. These blue spots under the microscope are isolated nuclei. We get hundreds of thousands of nuclei from just a few milligrams of brain tissue. Inside the nucleus, we measure biomolecules and the switches that regulate them to understand brain disorders like depression.